Samuel Miravet. I'm postdoctoral researcher in the group of Shinichi Sunagawa at ETH Zürich, and maybe most of you are familiar with the process of gene annotation of a genome, but you are not aware that this usually sets a threshold of 100 amino acids. This is mostly because if you don't include this threshold, a lot of small open reading frames that we know they are non-coding will be labeled as coding. So this doesn't mean that we don't have small proteins in a genome. In fact, there are plenty of examples and plenty of functions that can derive from these small peptides or small proteins. But the problem is that experimental approaches usually overlook them. During my thesis, I already worked with a model organism, one of the smallest free-living bacteria, and you can see that the number of small proteins that could be detected by different approaches was dramatically increased. But still, this is a challenge to annotate at large scale. So that's why with the HFSP, I'm trying to first develop an artificial intelligence approach to identify SEPs and improve annotations of different genomes, and I'm using as model of study the global ocean microbiome. Why the global ocean? This is one of the, well, it's probably the largest environment in the surface of the planet, and is also responsible for the 50% of the production of oxygen in the Earth. And 2/3 of the biomass of this environment is, in fact, microbes, that they play many important roles, like being the first link in the food chain, but also different metabolic diversity and different functions that come from the immense diversity of ecological niches that you can find in this environment. So one of the problems that I faced at the beginning of this fellowship, it was the fragmented data that you can find for this environment, so that's why I'm happy to introduce here our resource, microbiomics.io Ocean 2, which is an open source platform to navigate more than 12,000 different samples, collecting more than 300,000 different genomes from the ocean, which totals more than 5 million genes that we are searching, and this is not including small proteins. This is the next step. So from this resource that you can see there, the distribution in the map, we are planning to leverage different bioinformatic prediction tools to include everything on an artificial intelligence model to predict this from a new genome. We are also including different databases to also predict some of the interesting functions that we can collect from these small proteins, like signaling peptides or antimicrobials. And finally, we will try to address different fundamental questions like the role in the origin of larger genes, for example. So if you are interested in this, please visit me in poster 19. I think that's old, so probably it's 21 now. So that's all. Thank you very much.
Thank you very much.